Okay, so let's go to the next speaker. It's a pleasure to receive Dr. Daniel Brat. And I would like to thank in advance for accepting the invitation to join our annual symposium. So Dr. Brat received his MD and PhD from the Mayo Medical and Graduate Schools, and then completed residency in anatomic pathology and a fellowship in neuropathology at Johns Hopkins Hospital. And he further joined the Department of Pathology and Laboratory Medicine at Emory University. So since 2017, Dr. Brat is professor and chair of pathology at the Northwestern University, Feinberg School of Medicine, and pathologist in chief of Northwestern Memorial Hospital. So concerning his research interests, Dr. Brat directs a basic and translational research lab that investigates mechanisms of glioma progression, including the contribution of hypoxia, genetics, tumor microenvironment, and stem cells. So Dr. Daniel, thank you so much for being here with us and to share your parts of your data. And thank you so much. You're still mute.
Can you hear me?
Yes.
Better? Okay, well, thank you very much for the kind introduction. It's really a pleasure and an honor for me to be presenting here. I'm going to be presenting on the diffuse gliomas, which is my academic interest, my research interest. And the little niche that I like to add to is how these tumors progress from non-necrotic, non-hypoxic tumors to highly hypoxic, highly necrotic tumors. That's kind of the angle I take on these diseases. The family of diseases that we're going to discuss are the diffuse gliomas. They include those tumors that affect the central nervous system by infiltrating the CNS parenchyma by single cells that are neoplastic, shown here. All these individual angulated cells are neoplastic, and they go right through the neuropil of the brain, and in this case create some edema, fluid that flows into the brain in response to the injury. You can see that on the MRI shown here in the bright signal and the expansion of the temporal lobe, and all of that's in comparison to the normal brain, which has linear arrays of oligodendrocytes, scattered astrocytes, and a delicate but homogeneous and undisturbed neuropil. As I've said, the topic that I'm very interested in investigating and determining mechanisms have to do with the progression from a relatively non-necrotic, non-hypoxic environment where tumor cells are infiltrating almost as a homogeneous sheet shown here in pathology and here in the MRI. You can barely see the mass here, but over time, this tumor progresses to somewhat of a mushroom cloud, spherical mass that grows very rapidly outward and is ultimately fatal within a very short time. Almost all patients that have glioblastoma go through this phase that generates central hypoxia and necrosis. They also develop this rim of highly hypoxic tumor cells called pseudopalisades, a lot of angiogenesis here at the periphery, all in response to this central necrosis. So I'm going to talk a little bit about progression and its relationship to genetics, about the tumor microenvironment, including the immune cells, and what role glioma stem cells play in this evolution as well. I think the important thing to get straight right away is that high-grade gliomas, including the tumors that were once referred to as glioblastoma, includes multiple genetic entities. So here is a frequency distribution of high-grade gliomas that we all noted were trimodal as we were educated, but really didn't know why. Now, over the last 20 years, it's become clear that one genetic subtype, really a disease type, is called the IDH wild type gliomas, typified by glioblastoma, shown here, peaking in the 60s, whereas there's another subset of disease that are IDH mutant that occur more in the 30s and 40s, and then others that occur in the pediatric group that have histone H3 mutations or other types of genetic alterations that distinguish them. So today we're going to be talking almost exclusively about the IDH wild type diffuse gliomas, mostly glioblastoma, but it's important to realize that there are other family members as well that may have similar type mechanisms. And this has really been brought home to us over the last 10 years. If this is a analysis of 1,000 different diffuse gliomas, and this is a whole genome DNA methylation profile showing one subset that is relatively hypermethylated compared to one that is hypomethylated. And I think the key thing to realize here is that based on methylation profiling, as well as any other platform for analyzing these tumors like gene expression, proteomics, microRNA, copy number, mutational spectrum, these can be developed, subdivided very cleanly into IDH mutant and IDH wild type tumors among adults. And again, we're going to talk about the IDH wild type tumors. Now, first, if we just look at the genetic alterations that have been determined by the TCGA over the past 15 years and just lay them out in terms of frequencies, you can see that they fall into different categories, including those mutations that involve receptor tyrosine kinase pathway members, PI3 kinase pathway members, MAP kinase, P53, and retinoblastoma pathways. And each of these colored hashtags represents a mutation. So you can see that these are high mutation load type tumors, often including multiple genetic pathways. If we look at it a different way and break these mutations into just 3 pathways shown here, receptor tyrosine kinase, P53, and retinoblastoma, we can see that almost 100% of these IDH wild type glioblastomas have genetic alterations in each of these pathways. RTK typified by EGFR, but also P10 mutations and others. P53 by mutations in the gene itself, but also amplifications at MDM4, MDM2, and loss of CDKN2A, and retinoblastoma affected by, again, mutations of the gene itself, but also genetic alterations that include loss of CDKN2A/B. A fourth pathway that has become clear more frequently in primary wild type GBMs is TERT activation through mutation of its promoter that are activated. So in primary GBM that are IDH wild type, there are really 4 pathways that are almost uniformly altered. And I think that speaks not only to the treatment resistance, but also to the challenge that there will be in the future trying to target these by small molecules. That is, there's just multiple pathways that are activated, each of which can drive neoplasia. Also of interest from a molecular standpoint is that, if you just looked at that last slide I presented, you might think that these are relatively uniform in terms of their gene expression because those canonical genetic pathways are affected, but that's actually not true. And the combination of genetic alterations seem to give rise to different transcriptional families, such that if you take a large number of primary IDH wild type glioblastomas and do gene expression analysis and put them through a hierarchical clustering, you'll see that one subset is characterized by NF1 mutations and retinoblastoma mutations and have a mesenchymal gene expression signatures. Another typified by P53 genetic alterations and PDGFRA amplifications are the proneural type. And then there's the classical subtype that seem to have the signature of EGFR amplification and PTEN mutation, as well as some others. Now, we don't use these classes clinically. That doesn't mean that they're not important. And in fact, I would bet targeted therapies of the future will be directed at some of these differentially expressed genes or protein products, including things like immune profile genes or angiogenic genes and gene products, et cetera. Driving this home even further, if you look at whole genome methylation profiling, you'll also see that these tumors have multiple subsets within the IDH wild type group. So similar to gene expression profiling, there is a receptor tyrosine kinase family 1 typified by PDGFR-A amplification shown here. Receptor tyrosine kinase 2 typified by EGFR amplification, and mesenchymal typified by NF1 mutations and RB1 mutations. So methylation profiling, I think, is the new platform that is going to be one of the more robust in terms of molecular fingerprinting of different disease states. You can see how well methylation profiling is able to separate tumors that are very similar under the microscope. For example, the tumors that are IDH mutant are up here, and those that are IDH wild type are down here. For over 100 years, we could not tell the difference between these under the microscope, but methylation profiling has no problem subdividing them, and then even subdividing those tumors that are IDH wild type GBM. So can we use all of this data in a way that is more predictive and prognostic and can direct therapies in the future? We have all of these images based on MR scanning. We've got histology, we've got genomics, and we've got clinical data. How can we harness all of that data into predictive modeling that is much more sophisticated than under the microscope? Well, we, and this effort is led by Lee Cooper, who is the director of genomic pathology here at Northwestern now. We've attempted to use deep learning approaches to molecular data and then try to layer on to that some of the features that are captured in histology as well, and have been, I think, pretty successful in developing these models. So I wanted to show you at least, the titles and authorship of some of these papers that where we have used deep learning methods in order to first use just the genetics that were captured within the Cancer Genome Atlas, and using some of these methods to figure out which features are most predictive of outcome if you look at all of the diffuse gliomas that are included in that data set, which includes hundreds and hundreds of patients with glioblastoma. And if you use these methods, and again, you train the model on outcome and kind of a Cox survival model learning paradigm. And if you use this approach, you actually find that artificial intelligence can predict outcome based on genetics pretty well. And it suggests that among all of the genes that are altered in this disease, and also including all of the terms that get applied, CDKN2A or P16 deletion is the worst prognosis factor. Deletions on 10p are also very poor risk factors, and age at diagnosis. Those factors that are good prognostic features include IDH1 and 2 mutations if we combine those different disease sets, and SMARCA4 mutations. So we can use these evolving methods in order to filter through these immense data sets in order to tell us which factors seem to be most relevant to patient survival. We can also incorporate histology images from the same diseases in order to enhance our predictions. So again, if we think about trying to train a system to predict outcome based on histology and incorporating genetics, we can train the system based on outcomes and have the computer vision search through all of the features that are within slides and then incorporate genetics to predict outcomes even better. What we've shown is that even just using histology alone, the augmented intelligence approach shown here as SCNN does just as well, if not better, than humans in predicting outcome in terms of histology and grade. Whereas if we include some of the just basic genetic alterations as well as a modeling system based on digital pathology images, we actually can predict outcome the best, shown here in red. And I would encourage you to look at this work by Lee Cooper on predicting cancer outcomes in diffuse gliomas using convolutional networks. I think it is the way we will be progressing in the future in terms of diagnostics by incorporating both genetics, molecular profiling, and histologic features. So genetics takes us a long way in terms of describing progression, but there are other features that I think are telling within the diffuse gliomas that are much more biologic. As I've said, nearly all patients with high-grade gliomas go through a phase where hypoxia and necrosis develop centrally within the tumor, and then this tumor-like ball, if you will, grows radially in 3 dimensions within an enclosed skull that is not able to accommodate that growth. And that is why these tumors are so fatal in the short term after they've gone through this accelerated growth phase. Now, I've been interested in looking at this phenomena with the development of necrosis and the tumor, again, moving out in this direction in the plane that I've captured, but you have to realize it's doing that in all 3 dimensions as well. This tumor shows the hypoxic region directly next to the necrosis, the angiogenic rim shown here, but then the migration of tumor cells off to the periphery and overall expansion of this tumor. Now, I think there's a few ways to try to explain this growth. One, I think you could say, well, the tumor is growing so rapidly that it outgrows the oxygen supply and becomes hypoxic and necrotic centrally. I think that has been the prevailing dogma over the years. This is the so-called diffusion limited hypoxia and necrosis model, because the oxygen doesn't diffuse centrally enough to meet the metabolic demands. Another explanation is that the vasculature actually becomes compromised and we get perfusion limited hypoxia and necrosis. That is, a central vessel becomes compromised and the tumor cells directly surrounding it become hypoxic, necrotic, and migrate away or die. And it's actually this model that I favor as a neuropathologist. And I've suggested that the model is that a diffuse glioma grows till a certain point in time in cellular density, and then there is vascular occlusion that is associated with intravascular microthrombosis that leads to focal hypoxia, necrosis, migration of tumor cells away that set up angiogenesis to support further growth, and that this is the nidus of progression that carries through towards mortality. And I feel strongly about this because you can actually see these microthrombi within vessels, shown here, with the vessel becoming congested with blood cells behind that, and the development of these migrating cells or pseudopalisades surrounding the developing central necrosis. So this is the model that I've been putting forth for years. One avenue of research has been to investigate, well, what causes the thrombosis? And we've shown that the body's most potent procoagulant tissue factor, which is a protein, normally not expressed anywhere near the vasculature because it's so potent, is dramatically overexpressed within gliomas themselves in response to the accumulation of specific genetic alterations. And when the bloodstream becomes more permeable during the disease progression, tissue factor outside the vessels becomes exposed to factors within the vasculature to start this predominantly fibrin-induced clot that is seen within the vasculature of high-grade gliomas. So we think that the development of hypoxia necrosis is caused by vasoocclusion, the tumor growing in a manner that compromises its blood supply through the induction of small microthrombi that then cause hypoxia-induced glioma progression. So shown at a little bit more detailed level here is a vessel in the middle of a glioma that becomes thrombotic. Tumor cells migrate out. There's an accumulation of stem cells that we'll talk to, and an influx of microglia that become activated, and monocytes exiting the vasculature to become activated macrophages. So it's this phenomena that induces tumor outgrowth, including the angiogenesis that's induced by these hypoxic cells, leading to the tumor microenvironmental expansion of these highly fatal tumors. So I've talked about tumor-associated macrophages a bit, and I think all diseases of the brain that damage tissue have an influx of macrophages, monocytes that develop within the bone marrow and are circulated in the bloodstream, exit into the brain in response to damage. That happens in stroke, that happens in traumatic brain injury, contusions, etc. And brain tumors are no different because they're a damaged tissue, if you will, as well. However, in diffuse gliomas, when macrophages come in, they convert themselves from a M1 state or M1-like state that is really meant to, create an inflammatory environment that's necessary, where they have appropriate immune function and suppress the tumor to an M2 state or M2-like state. Those are old terminologies where everything from angiogenesis to tumor cell migration to stem cell development to immunosuppression is supported. So we're very interested in why these macrophages convert from an M1-like state that's inflammatory to an M2-like state that's promoting the tumor. There's been obviously a lot of interest in this area with the development of checkpoint inhibitors, et cetera, in understanding the interactions between T cells, macrophages, and glioma cells, and we're among those that are interested as well. Just to show this schematically, what happens from the time when the tumor does not have any hypoxia and necrosis shown on the left, there's a rim of flattened macrophages that surround blood vessels within the diffuse gliomas. There are also scattered, modestly activated microglia that get picked up by staining. And that's, it's not normal. There is some activation of microglia and potentially a few marrow-derived myeloid cells that creep in. But once the development of necrosis and hypoxia set in, you can see all of these perivascular macrophages stained in brown for CD163 have become activated. Cells that were within the vasculature come into the brain parenchyma, and by the time the tumor is fatal, nearly 35 or 40% of all cells within the mass are accounted for by activated tumor-associated macrophages that, again, have converted to an M2-like state that is tumor promoting rather than inflammatory and opposing the growth of the tumor. So this is exactly the phenomena that we are interested in studying and, potentially using tumor-associated macrophages to combat the tumor as would be appropriate. So why does this really matter? Well, we think it's going to be important therapeutically. I think one thing that we all hope for is targeted therapies in the future, and some of this may be dependent on the immune status of the tumor or tumor type at least. So here we've looked at 3 of the different transcriptional subtypes of glioblastoma. And we see that there's different densities of tumor-associated macrophages within the substance of the brain tumor, with the mesenchymal subclass having by far the most, the classical subset having second, and the proneural having the least. Now, if we look in the TCGA data at survival, we actually even see something very different. We find that within the mesenchymal GBMs, those tumors that have the highest level of TAMs actually have a slightly longer survival compared to the low. But if we look at the proneural, those tumors that have the highest level of macrophages actually have the shorter survival. So this is going to be very context and tumor dependent in terms of prognosis and the meaning of these TAMs in the setting of different genetic subsets. But it's also becoming clear that potential therapies may interact differently with subsets that have different genetic and macrophage content. So in this case, we see that a specific form of immunotherapy based on dendritic cell vaccination strategies are much more effective in those tumors that are mesenchymal and have all of those macrophage infiltrates than in others. So very interesting how this may be playing out in terms of differential response to immunotherapies as we learn more. One of the approaches that we've taken to model these diseases in vitro includes neurosphere and cerebral organoid models. Now, of course, we eventually want to get to in vivo modeling in mice that correlates with the human disease, but the in vitro opportunities are growing more and more complex, and we're able to, I think, more faithfully mimic the human disease in vitro as we start to use neurosphere and cerebral organoid models. The advantage is that they do have a matrix that's similar to brain and the tumor microenvironment of gliomas. If you have the right brain tumor models, you can incorporate different cell types and differentiate them within the organoids and neurospheres in order to manipulate, and you can manipulate the cells and the tumor microenvironment by exposing them to hypoxia, incorporating different genetic alterations, if you wish, cell densities, etc. There are some challenges associated with this in that you need live imaging in 3 dimensions in order to capture things like glioma invasion through an organoid or microglia infiltration within an organoid or neurosphere. But we started to make some up some ground on this, and one of your fellow country folks, Tatiana Lobo, has been working with us for some time, along with Cheryl Olson, on developing these models. And I just want to show you how you can use these. So this is a neurosphere model that uses GBM cells derived from a patient and grown in serum-free media, where the cells will aggregate into neurospheres. They have the genetic alterations of the native disease. They have the capabilities of forming stem cells that can be used to studies. And we've labeled microglia in this case and exposed this culture of neurospheres to these microglia that have been labeled with this magenta. And we can study them live in 3 dimensions as they infiltrate the tumor. And we can also do this in normoxic and hypoxic conditions. And we're getting the sense that hypoxia, in fact, increases the amount of microglia infiltration into these neurospheres. We can, we can do much more precise modeling of speeds and trajectories of these cells going into a neurosphere using this type of in vitro model than we can ever do within an in vivo model. We can, again, alter the constituents of the media. We can alter the hypoxic environment or study the effects of hypoxia within the neurosphere itself as it affects this migration. So using a neurosphere, and then we can also use organoids as a model. And of course, organoids have use neural stem cells that can differentiate along multiple different lineages, including mature neurons, neuronal stem cells. We can differentiate cells into glia and make a very complex system that comes close to having the same environmental features of the normal brain. So shown here with the DAPI stain in blue is an organoid, very large in this case and complex, and we can incorporate human glioma cells into it. In this case, we have GFP labeled NU1485 cells, and using this system, we can study migration properties. We can expose to hypoxia, we can add in microglia as well, and try to mimic glioma invasion much more reliably. And here you can see that Tatiana has captured the same system essentially at 2 different times, one at 3 days and one at 6 days, showing how much proliferation and infiltration there is of the glioma into this neurosphere that we can then go on to manipulate and study much more in detail. And again, we can also incorporate labeled microglia into this system, which you can see here, and study invasion as it relates to many environmental factors, including hypoxia, which we can track with hypoxia probes, like is shown here. So again, we are developing these models thanks to Alison Moultrie at UCSD, and Tatiana has been extremely important in leading this effort within our lab, and we think it'll be very fruitful for the future. Now, we can also try to model these microenvironmental events in vivo. It is more challenging, but it also, I think, will be rewarding in the long run. So in this case, we want to model the development of microthrombosis within a glioma to study the downstream effects of hypoxia and necrosis in a mouse model. So first we inject a tumor, and we can do that either through directly injecting the human cells, human tumor cells, or we can use an RCAS TVA model in which we incorporate the avian virus that packages oncogenic alterations into the brain of an animal that's expressing the avian virus in a specific subset of normal cells. And we have both models, and we allow the tumor to grow. We put on a skull window, we replace a part of the skull with a glass cover slip, and then we give this agent called rose bengal, shown in red here. Rose bengal gets into the bloodstream, and we can photoactivate that with a laser in a very specific point within the blood vessel, and that causes focal thrombosis. And then we can study the events that occur post photoactivation live and at various time points. And again, this is what this looks like for real and not schematic, shown here using the mouse basically as a microscope platform and being able to visualize through 2-photon microscopy the dynamics that are occurring within the tumor itself. And in this case, we have a GFP labeled tumor shown here at low magnification. And we can see the blood vessels streaming through that tumor because they are actually labeled with rose bengal, which is red. We can go to higher magnification and actually target a vessel by activating it with the laser and cause focal vascular defect. And we're getting pretty good at that and disturbing the blood vessel at a very focal location and being able to see in very short time period the extravasation of, in this case, a GFP labeled macrophage through the bloodstream and into the tumor. And that is the goal. So we did not have tumor cells in this case. We were just monitoring the ability of macrophages to get out into the bloodstream, get out of the bloodstream into the brain. I'm not sure if this is working or not, but there you can see that green dot is actually live photoactivation of the rose bengal. This is a 3D reconstruction where you can see the focal deficit. And here you can see the first example of microglia that are in the circulation here getting into the brain parenchyma. And similarly, if we wait for overnight after this photoactivation, we see just a large number of macrophages coming out into the tumor microenvironment. So we have the models set up to study live events, both in organoids in vitro and within mouse models in vivo. And again, we are very interested in the tumor microenvironment in terms of hypoxia, necrosis, and the influx of macrophages that become immunosuppressive. But we're also interested in how glioma stem cells emerge within this altered tumor microenvironmental state. Now, glioma stem cells are a critical component of high-grade gliomas. There's no doubt about that. The subject matter is a bit murky sometimes because we use markers that aren't perfect and not uniform across different labs throughout the world. But I think we're gaining some better understanding of the dynamics of glioma stem cells as they exist and what happens under hypoxic conditions. So some of the things that I think most people hold true about glioma stem cells, no matter what lab is studying them, is that they tend to localize to specific niches. We'll talk a second about that. They're biologically potent and therapeutically resistant. That is, the initial papers said that if you use glioma stem cells, a tumor can be initiated with 100 of those cells. Whereas if you try to start a tumor in an immunocompromised mouse with 10,000 cells that don't have stem cells, you'll be unsuccessful. And one of the features that we are very interested in is asymmetric cell divisions, which are an absolute necessity for a cell to be considered a stem cell. That is, the cell must be able to self-renew to be able to continue its stem cell state, and it must be able to differentiate in a multi-lineage capacity, as shown here for normal neural stem cells. So we do know that glioma stem cells, if you recognize them by CD133, which is one of the gold standards in neuro-oncology, they do tend to cluster in two specific niches. One is the perivascular niche shown here. And again, here's a vasculature going through this neoplasm. And here are the glioma stem cells in the perivascular niche. And the other niche is right around necrosis in this hypoxic microenvironment. And this immunofluorescent image is also showing the accumulation of CD133 positive cells around central necrosis in the hypoxic environment. And I think everybody that studies stem cells does agree that if you culture these cells in hypoxic conditions, you also enrich for both CD133 and the capacity of these cells to functionally be measured as stem cells. So again, since this specific feature emerges with glioma progression, the development of hypoxia and necrosis, and it doesn't exist in the state before that, we are very interested in how this phenomena gives rise to an enrichment of stem cells and how that phenomena becomes critical in radiation and chemotherapy resistance of the tumor as a whole. We have done some preliminary studies on some of the factors that might give rise to therapeutic resistance, and for example, have looked in single cell data and bulk tumor data from the GLASS consortium, where we were able to correlate the level of hypoxia with the expression of BCL2, and found that there was a pretty strong relationship between BCL2 and the degree of hypoxia. And we were able to go on and show that, in fact, it was hypoxia in the in vitro environment was highly associated with the upregulation of BCL2 in a manner that you might predict would predict therapeutic resistance to alkylating chemotherapy or radiation therapy. There is a hint that the CD133 positive hypoxic tumors might have higher levels of BCL2 than the non-CD133 expressing cells, but we need to do more experiments to see if that trend holds. But clearly there is a big question in the field about what cell population under what conditions gives rise to therapeutic resistance, whether it's a state of stemness or a state of senescence or quiescence. And we intend to get at those questions. But in the immediate term, we're very interested in the phenomena of symmetric versus asymmetric cell division, because we think that is such a central phenomena that distinguishes many stem cells from non-stem cells, number one. And we think that the process of symmetric versus asymmetric cell division becomes altered in the neoplastic state in a manner typically that favors more symmetric division or more stem cell presence with a neoplastic disease. So in symmetric cell division, you can imagine that a mother cell becomes just like two daughter cells. They're identical, and in fact, one stem cell can give rise to daughter cells. Asymmetric cell division, on the other hand, the stem cell mother cell has cellular cell fate determinants that are localized to one cell only, and the cell division is non-symmetrical and gives rise to one daughter that continues to be stem and one that can go on to differentiate. Well, these phenomena have been studied extremely well in Drosophila, okay? And you are, I'm sure, aware that Drosophila is a wonderful model for studying genetics and pathways that are preserved in humans and human diseases. And Subhash Mukherjee has been the one carrying out these investigations over the last 7 or 8 years in the lab. So, if you can think about cell division, asymmetric cell division being broken into the basal cell and the apical cell, with the apical cell maintaining stemness and the basal cell going on to differentiate, there's multiple elements that have to fall in place to activate Notch to create stemness, and also included in there are Wnt signaling. Whereas on the cell differentiated side, you have to inhibit Notch as the final common pathway. And there's a complex involved with this regulation that includes Prospero, Miranda, and Brat. Brat has to be, happens to be my last name. I've been very interested in that gene and gene product. And we've studied the mutants in that Drosophila model that give rise to a brain tumor because of altered asymmetric cell division. Now, what's that look like? Well, if you have a fly that has a mutation in the gene Brat, the brain specifically becomes extremely enlarged. So this is one lobe of the brain, this is the normal counterpart, and you can see that it's extremely enlarged compared to the normal at the larval stage. This brain tumor is completely full of undifferentiated neuroblasts. They have lost the ability to differentiate, and they only undergo symmetric cell division. So this is an extremely valuable model to study symmetric versus asymmetric cell division and the pathways that go awry. Importantly, if you take cells from this expanded brain tumor and put them in an adult fly, the tumor will continue to grow. So it's a malignant-like proliferation of neuroblastic cells that have lost the ability to undergo asymmetric cell division. Well, we've been very interested in studying the human correlates of this and to define the pathways involved first in Drosophila and use genetic screens to find out how to counteract this that might be viewed as developing targets for future therapies. So Subhash developed not just a larval model, which has limitations, but he developed an adult brain tumor model of Drosophila Brat, where he introduced RNAi developmentally and could develop this brain tumor model in flies that we could use as a genetic screen. And here you can see with the tubulin cherry staining that there is a huge population of neuroblasts in this yes, there are some parts of the brain that are expressing neuronal markers, but in the normal adult brain, there are no neuroblastic markers left. So we're very interested in this neuroblastic cell population that main stem cell properties. We've characterized these by showing that there are in fact stem cell markers like MIRA expressed in these cells, that they are more proliferative and they express activated Notch. So they have the properties of stem cells when you knock down Brat in an adult fly. Importantly, we can also use these models in the brain or in the eye shown here in order to do genetic screens to see which genes, when knocked down, counteract the suppression of Brat. Again, potentially providing a target for therapy. So here's the control eye, and we use the eye because you can screen it without dissecting the brain. You can just see how the architecture is normal here and very abnormal once you use Brat RNAi. And then what we found is if you knock down P35 or its binding partner CDK5, you can actually restore the normal microanatomy of the eye, thereby counteracting the effects of Brat RNAi within this system. And we think this provides a potential therapeutic target for aberrations of asymmetric cell division. Now, and again, here we show that activated CDK5 is expressed precisely in those cells that are stem cells and could be a target of therapy, and is co-localizing with the apical cell during the asymmetric cell division process. Importantly, there is a homologue of Drosophila brat in human. It's called TRIM3, and here you can see that it has a decent amount of homology with the fly gene. In the human, it is located on chromosome 11p15.5, and it's deleted in a large subset of GBMs. Importantly, it is a tumor suppressor that impairs growth, attenuates stem cell properties, and favors asymmetric cell division in human gliomas as well. And we've shown that because TRIM3 is normally suppressed in human gliomas in its natural state. And when we overexpress it, we find that you actually suppress the amount of stem cells that are present. So it's a differentiation marker, if you will. And when you overexpress TRIM3, you actually knock down the amount of activated Notch within the nucleus of these cells, shown here, and your ability and the ability of Notch to express its target genes like HES1. So what we found is that a gene that controls stem cell behavior in flies is also important in regulating stem cell behavior in humans. And we think that we could potentially target similar genes that are targeted in Drosophila to reverse that, like P35 or CDK5, which are also upregulated in these tumors. So what Subhash has gone on to do is to use an inhibitor of CDK5, and he's shown that he is able to, number one, inhibit the enzymatic function of CDK5, that this does have an effect of tumor suppression in vitro, and he can do limiting dilution assays and show an effect on glioma stem cells in vitro as well as targeting specific markers of stemness within glioblastomas in vitro. And we're obviously very interested in pursuing these types of therapies to target glioma stem cells in the future. We think it's a wonderful model to start with Drosophila, identify those molecules that control stem cells and asymmetric cell division, and then find the homologs in the human disease and target them therapeutically. So hopefully in today's lecture, I've told you a little bit of my view through the lens of a neuropathologist interested in glioma progression, which includes considerations of genetics, which includes the tumor microenvironment and the influx of myeloid-derived cells as tumor-activated macrophages, and the role of glioma stem cells becoming enriched in the hypoxic environment and having aberrant asymmetric cell division. So I would like to thank everybody in my lab, as well as collaborators throughout Northwestern and previous institutions. And with that, I think I'll thank you and take any questions.
Okay, so Daniel, thank you so much for your excellent talk. I think we had a very nice overview of your current research interests. And I have a question from the chat here. It came from Luciana Barros. Excellent presentation. in your hypothesis, blood vessels lose their function when the tumor starts? Do you have any hypothesis why it happens, or what factors lead to this loss of function? A second question is, if this impairment on vessels damper immune response, preventing leukocytes to migrate into the tumor or glial cells to function properly. Can you see the chat here?
Let me see.
I think it's easier reading than I.
Okay. Okay, so let's talk about the vessels losing their function first. So the important thing to recognize is that we don't know about a patient having a brain tumor until they develop symptoms, right? We don't screen for these. So we usually catch brain tumors at a late state of their progression. So we never capture them at the beginning in reality. Now we do capture some of them before they become hypoxic and necrotic. And that's when I think the vessels lose their function. It's the patient has had their tumor for quite a while, growing over the course of, who knows, months at least, in some disease types, years. And then there's a click where I do think that the blood vessels become dysfunctional. And what happens, I happen to think that the coagulation system, so for over 150 years, it's been known that there's a link between cancer and coagulation. Trousseau first described it that patients with cancer, in particular pancreatic cancer, but glioblastomas as well, have a very high rate of deep venous thrombosis and pulmonary emboli. That's actually driven by tissue factor from the tumor cells themselves getting into the circulation and causing that effect systemically. I happen to think in the tumor itself, not even getting into the systemic circulation, the expression of these coagulation factors, chief among them tissue factor, causes microscopic thrombosis. So it's Trousseau's phenomenon that happens systemically, happening very focally within the tumor microenvironment. So that's what I think is happening. A second question, if it is impairment on vessels, dampens the immune response, preventing leukocytes to migrate to the tumor cells, glial cells, if this? It could. So these events that occur are very destructive once they get started. The pre-hypoxic, pre-necrotic glioma is not destructive to the extent that glioblastoma is destructive. Once hypoxia and necrosis get established, the vasculature is extremely leaky, okay? And immune cells can get past that very readily, which I showed. So I don't think it dampens the immune response. I think once the disease has progressed, it actually allows the immune response to occur. But the immune response, yes, it's not appropriate. So the tumor itself has an immunosuppressive effect on its environment, on the immune cells. And that phenomena, I think, is not as well understood, at least by me. That's very complex, but we're trying to figure out why that is and what can help reshape that.
Okay, so I have two questions. So the first one is that there is a recent data have shown that podoplanine may play a role in glioblastoma-associated thrombosis. So do you have any evidence that, so in this protein is very important to activate platelets. Do you have any evidence that you have platelets in the glioblastoma microenvironments?
I don't. I don't have that evidence myself, and platelets versus fibrin, they're both included in most clots, but differentially depending on the clot type of clot that forms. I know the literature about protoplanin, but I haven't done the experiments myself. And, you know, platelets in general are an intravascular cell type, if you will, a nucleated cell type. And they're, you know, they can extravasate into the tumor microenvironment. I typically think of them being most functional within the blood vessels, but I'm sure there's a lot that we don't know about their function as they extravasate out as well. So could be extremely significant. I just don't have that data.
So the second question is, there is some data have shown a correlation between stem cell properties and tissue factor expression. So do you have any evidence that the TRIN3, I guess, correlates with tissue factor expression in your models?
Again, great question. And yes, I find it very fascinating that, you know, these events seem to occur in the perivascular niche where all these cell types seem to aggregate and where there is tissue factor expression. So many questions to answer, so little time, you know, so few people to work on the questions. But yes, I'm interested in the interface of TAMs in the perivascular environment and glioma stem cells, as well as in the necrotic niche. So yeah, I think the localization is very curious, the co-localization, and wonder about, you know, direct or indirect effects that one could be having on the other.
Okay, so I would like to thank you once again for joining our annual symposium. And I want to thank you again for the excellent talk. And that's all. So thank you so much.
Okay, well, thank you. And everybody have a very nice symposium. And thank you very much for inviting me. It's been an honor.
Thank you so much.
Bye-bye.
Bye-bye.